Assalamu alaikum. In this video, we are going to talk about the cellular basis of the immune response. And under this topic, we will cover several subtopics such as the origin of the immune cells. Then we will cover the different types of immune cells in great detail, and we will talk about what are the immune cells and what are their types, their activation, their regulation, and their functions. First, in this video, we are going to talk about the origin of immune cells. So, the capability of responding to immunological stimulus rests mainly with the lymphoid cells. During embryonic development, blood cell precursors originate mainly in the fetal liver and the yolk sac. These are the main areas where the blood cells originate in the embryonic life. In postnatal life, after birth, the stem cells reside in the bone marrow, and the stem cells differentiate into cells of the erythroid, myeloid, and the lymphoid series. So first of all, we have stem cells, and the stem cells that are differentiating into different blood cells are called as hematopoietic stem cells. Hematopoietic stem cells are multipotent stem cells, and they have the ability to differentiate into a number of cells. So the hematopoietic stem cells differentiate into the erythroid, myeloid, and lymphoid series, and the B cells, T cells, and natural killer cells originate from the lymphoid series, and the macrophages, they originate from the myeloid series. So the lymphoid solid give rise to T lymphocytes, B lymphocytes, and natural killer cells, and the ratio of B cells to T cells to B cells in the body is 3:1. There are 3 T cells in comparison to 1 B cell. Now let's move towards some of the details of T cells, B cells. The T cell precursor differentiate into immunocompetent T cells within the thymus. Thymus is a lymphoid gland that is situated in the thoracic cavity above the lungs. So the T cell get mature in the thymus, and that's why they are known as T cells, because the T stands for thymus. Stem cells lack the receptors for recognition of the antigen, and they also lack other glycoprotein receptors or the molecules that are present on the surface of the T cell, which include the CD3, CD4, and CD8 molecules. But during the passage of T cell through the thymus, these receptors are created, or these are produced on the surface of the T cells. The stem cells, which lack both the CD4 and CD8 cells, they are called as double negative cells because they neither have CD4 nor CD8. But they first differentiate to express both of these receptors on their surface, and those cells that contain CD4 and CD8, both receptors or both molecules on their surface, they are called as double positive T cells. Then these double positive T cells further differentiate into single positive T cells. Either they contain the CD4 receptors and they become the helper T cells, or they contain the CD8 receptor on their surface and they become the cytotoxic T cell. Both the double negative cells and the double positive cells are present in the cortex of the thymus, and the single positive cells, either with the CD4 or CD8 on their surface, they are present in the medulla of the thymus. Within the thymus, two very important processes called the thymic education occurs. First is those cells which are CD4 positive or CD8 positive cells bearing antigen receptors for self proteins, they are killed, and this process is called as colonal deletion, and this is achieved by a process called as programmed cell death, otherwise known as apoptosis. The removal of these self-reactive cells, a process called negative selection, results in the tolerance to or on proteins, and the significance of this process is that it prevents from autoimmune disorders. The next process that takes place in the thymus is that the CD4 positive cells and the CD8 positive cells that do not react with the self MHC protein major histocompatibility complex are also killed. This results in a positive selection for T cells that react well with the self MHC proteins. These two processes produce cells that are selected for their ability to react both with the foreign antigens via their antigen receptors and with the self MHC protein. Both of these features are required for an effective immune response by the T cells. During the passage of T cells through the thymus, each double positive T cell synthesizes a different highly specific antigen receptor. The antigen receptor is composed of different domains, and these domains include the variable region, the diversity region, and the joining region, and all these domains are encoded by different genes, and the DNA rearrangement between those parts of the DNA that contain those genes that results in the formation of a diversity of antigen receptors. Now let's talk about the B cells. B cells do not pass through the thymus. So the precursors of the B cells mature somewhere else, and the region where they get mature is called as gut associated lymphoid tissue, which is also abbreviated as GALT, GALT. And the other name for the gut associated lymphoid tissue is the Peyer's patches. Gut associated lymphoid tissue is the mammalian equivalent of the bursa of Fabricius in birds. So the B cells in birds, they get mature in the bursa of Fabricius, but in mammals the B cells get mature in the Peyer's patches, which are also known as gut associated lymphoid tissues. So that's the reason the B cells are called as B cells, because the B stands for bursa. B cells also undergo clonal deletion of those cells bearing antigen receptors for self proteins. This a process that induces tolerance and reduces the occurrence of the autoimmune diseases. But the site of clonal deletion of B cells is uncertain, but it is said that it is not the thymus. Natural killer cells. Natural killer cells are large granular lymphocytes that do not pass through the thymus, and they do not have an antigen receptor, and they do not bear the CD4 or CD8 molecules on their surface. In contrast to the T cell, B cells, and the natural killer cells, which differentiate from the lymphoid stem cells, macrophages arise from the myeloid precursors, the myeloid series. Macrophages have two important functions. First is the phagocytosis, and second is the antigen presentation. They do not pass through the thymus, and they do not have the antigen receptors on their surface. They bear class 2 proteins, class 2 MHC proteins, which is called as major histocompatibility complex, and this protein play an essential role in the antigen presentation to the helper T cells. In the next video we will talk about the T cells, their types, their activation, their regulation, and all the other aspects of the T cells. Allah Hafiz.